students, let us begin with the new chapter, Biomolecules. It is your unit 14. So, as the name suggests, you can understand that these are the molecules which are available in living organisms and how we define them. All living organisms are composed of several non-living substances. That is something very interesting, that we all living beings are made up of non-living substances. But the speciality of these non-living substances is they are arranged in a special manner due to which they are able to produce life. So biomolecules are complex, lifeless organic substances which build up living organisms and are required for their growth and maintenance. Now interestingly, most of the biomolecules, they are polymers. For example, you have starch. Starch is a polymer of sugar, normal sugar or carbohydrate. Proteins are polymers of amino acids. Nucleic acids are polymer of nucleotides, like that. Now let's begin with carbohydrates. These are naturally occurring organic compounds. So what does it contain? Carbohydrates must contain carbon, hydrogen, and oxygen, and they are primarily produced by plants. As you know, carbohydrates are our major um, ingredient in our food, and carbohydrates can be produced only by plants. These contain the polyhydric aldehydes, ketones, and large polymeric substances which can be broken down into polyhydric aldehydes or ketones. Now, what is the meaning of this? So carbohydrates, they contain lot of OH groups. That's why we call them polyhydric. And they have a major functional group which could be either aldehyde or ketone. The most common sugar which we use in our day-to-day life is sucrose, having the formula C12H22O11. Most of the carbohydrates, they can be represented by the general formula CXH2OY, where X and Y may be same or may be different. You can see the example of glucose. In the example of glucose, what we notice here, you can see glucose is having formula C6H12O6, which can be written as C6H2O6. Sucrose is C12H22O11, which can be written as C12H2O11. That means these are obeying the general formula CXH2OY. But there are certain carbohydrates which do not obey this formula, and one such example is rhamnose. You can see rhamnose is having having a formula C6H12O5. So here you cannot take out any number common to have H2O as one unit. Okay, so rhamnose is a carbohydrate, but it doesn't obey the general formula. Again, on the other hand, there are certain compounds which do obey this formula, but they are not carbohydrates. You can see here the example is acetic acid. Acetic acid molecular formula is C2H4O2, and you can easily write it as C2H2O twice. But we all know acetic acid is not a carbohydrate. Now, one important aspect of carbohydrate is they must have at least one optical center. By one optical center, what we mean? They must have at least one chiral carbon. Okay. So let's go to the classification. Based on their behavior on hydrolysis, they can be classified as monosaccharides, oligosaccharides, polysaccharides. Now what does that mean? Mono, as the name suggests, you can understand, monosaccharide means they have one sugar unit. That means those carbohydrates which, when you hydrolyze, do not give you a simpler form, such carbohydrates are called monosaccharides. That is, they contain the simplest unit of sugar. You can see it's written a carbohydrate that cannot be hydrolyzed further to give simple units of polyhydroxyaldehydes or ketones is called monosaccharides. We have a lot of examples: glucose, fructose, galactose, ribose. These are all monosaccharides. Then what are oligosaccharides? These are a collection of various carbohydrates. Those carbohydrates which upon hydrolysis give you 2 to 10. You may see 2 to 10 monosaccharide units upon hydrolysis. Such carbohydrates are called oligosaccharides. And we are using sucrose, which we are taking. It is a disaccharide. That means upon hydrolysis, it is capable of producing two simple sugar units. And you can see here raffinose is a trisaccharide. Raffinose is a trisaccharide. That means if you hydrolyze it, it will give you three simpler units. Then comes polysaccharides. So as you know, the name suggests poly means many. So those carbohydrates which upon hydrolysis can produce a lot of sugar molecules, simple sugar molecules. Such carbohydrates are called polysaccharides. And the common examples are starch, glycogen, cellulose, gums, etc. So that's how we can classify the sugar carbohydrates based on their hydrolysis. Now, next move to the other category of classification, and that is based on their reducing nature. When we did aldehydes and ketones, you must have learned that aldehydes and ketones, um, aldehydes can get easily oxidized by mild oxidizing agents such as Tollens reagent, Fehling solution, etc. Whereas ketones cannot be hydrolyzed easily. Now you know that most of the carbohydrates they contain either aldehyde group or ketonic group. But interestingly, whether they are aldehyde or ketone doesn't matter. Carbohydrates, they are capable of reducing both aldehydes and both Tollens reagent and Fehling solution, irrespective of whether they have aldehyde group or ketonic group, if they are monosaccharides. All monosaccharides, irrespective of aldehyde or ketonic group, can do so. So those carbohydrates which are capable of reducing Fehling solution and Tollens reagent, they are called reducing sugars. All monosaccharides are reducing sugars. On the other hand, those which are not capable of reducing Tollens reagent and Fehling solution, such sugars are called non-reducing sugars. So you can see the examples you have are, you have maltose and lactose. These are non-reducing sugars. No, maltose and lactose are reducing sugars. Sorry. So let's begin with the monosaccharides. Under the monosaccharides, we have to do glucose in detail. So if a monosaccharide contains an aldehyde group, you can see if a monosaccharide contains an aldehyde group, then we call them aldose. And if they contain a ketonic group, then we call them ketose. So depending on the number of carbons present in the monosaccharide, they can be termed as triose, tetrose, pentose, hexose, etc. Now since we have to discuss glucose in detail, so what is glucose? Glucose is an aldohexose. That means it has an aldehyde group and it has 6 carbons. Now what is the source of glucose in nature? Ripe grapes contain about 20% glucose, and in combined state, glucose is present in di and polysaccharides such as maltose. Maltose is a disaccharide of glucose, and starch, which is a polysaccharide of glucose. Now, how do we prepare glucose? Glucose, we have two methods to learn to prepare. In the lab, glucose is prepared by hydrolysis of cane sugar, that means the sugar which we take, in the presence of alcohol using dilute HCl or H2SO4. Now, glucose and fructose are formed in equal amounts. Glucose, being less soluble in ethanol, crystallizes out. So, that is the purpose, to use this alcohol in the solvent. Now, when sucrose is hydrolyzed, you get equal amount of glucose and fructose. Both have same general formula. Both are C6H12O6, but you cannot write two molecules of C6H12O6 because the compounds are different. Now, what is the purpose of using here alcohol? Because out of glucose and fructose, fructose is more soluble in alcohol and glucose is less soluble. So, since glucose is less soluble, it will try to separate out and it will be easy for us to separate the mixture. In most of the cases, there is an enzyme used here that is called invertase. Now, why the name of this enzyme is invertase? Because sucrose, when we check the optical activity of sucrose, sucrose is found to be dextrorotatory. And glucose is also dextrorotatory, but fructose is levorotatory. Now, the rotation of fructose is more than that of glucose. So, finally, what happens when you begin with sucrose, which is a dextrorotatory? After hydrolysis, when it forms a mixture of glucose and fructose, fructose being levorotatory and is having more optical rotation, the final solution become levorotatory. So there is an inversion in the rotation, and that's why the enzyme used out here is called invertase. And the whole process is called inversion of sugar, and this mixture is called invert sugar. And the other method from starch. Starch, the formula for starch you must remember, it is quite often asked in the Viva also. Starch is C6H10O5 whole thing n times. When hydrolyzed with water in presence of nitric acid and at more or less 2 to 3 bar pressure, not very high pressure, then we get n molecules of sucrose. So definitely starch is a better source of glucose because it gives you 10% glucose. There is no fructose formed here. So next topic will be structure of glucose, which we'll take up in the next audio.